focus of this video is on transcriptional regulation. We're going to talk about the main ways that prokaryotic cells and eukaryotic cells regulate gene expression at the level of transcribing DNA to make RNA. But before we can do that, we actually need to spend a little bit of time talking about gene organization, because it turns out that the way that prokaryotes and eukaryotes organize their genes within the genome is a little bit different, and that has some consequences for how they regulate their genes. So we're going to start with prokaryotic cells and just lay out some fundamental features of how prokaryotes' genomes are organized in general. So if we look at the DNA in prokaryotic organisms, what we would find if we looked at the genes and how they're arranged is that we'd have what's called high gene density. And what that means is there's not very much space between individual genes within the genome. They're pretty much clustered back to back within the DNA. So you look at, let's take a bacterium, a little rod-shaped bacterium here with one circular DNA molecule. We've talked about these before. We know that prokaryotes in general have one circular chromosome. And within that chromosome, if we take an organism like E. coli, what we would find is about 4,000-ish genes along that chromosome, each gene representing one instruction to the cell. This is something we talked about before, how to make one functional product, usually a protein. And if we looked at that DNA and those genes within that DNA, what we would find is that they are pretty much back to back to back with no sort of non-coding genetic sequence in that DNA molecule. That's what we mean by high gene density. If you look at any given region of DNA, you'd find a lot of genes clustered there and not much empty genetic information or unused genetic information. In addition, in prokaryotes, we see something interesting in that functionally related genes, that is, genes that code for proteins that are acting in the same sort of metabolic pathway, tend to be clustered together within the genome in units that are called operons. So clustering of genes that have some related function is found in prokaryotes, and those units are called operons. And importantly, within those operons, these clustered genes are all regulated together. Right? So if one gets turned on, they all get turned on. If one gets turned off, they all get turned off. So if we looked at one of these genes, pulled it out, and took a look at its structure in more detail, we'd see something. We took one of these operons and pulled it out of the genome and looked at it in more detail. And this would go on in both directions on either side. So you might have a set of genes. Let's just call them genes A, B, and C. Doesn't really matter what their particular function is. What's important is that these genes code for products within the cell, and as I said, usually we're talking about proteins here. They code for proteins that are functionally related in some way. They're operating in the same metabolic pathway. Right? They would be clustered right next to one another in the genome, and their transcription would be directed from a common, what's called a promoter sequence. Now the promoter sequence is the sequence that's found at the beginning of every gene that's transcribed, and it serves as the binding site for RNA polymerase. RNA polymerase is an enzyme that we talked about previously. It's the enzyme that copies DNA to make RNA. So the promoter is found at the beginning of the gene, and it serves as a sort of recognition signal for RNA polymerase to know where the gene begins. But in prokaryotic organisms, we would see genes clustered together, all served by one promoter. And so as RNA polymerase goes along and transcribes this bit of DNA, we would end up with one long mRNA containing the coding sequence for all of those genes. That mRNA would then be translated. We know how that occurs. We talked about that before, right? These would be ribosomes that would come along and bind to the mRNA, and through the process of reading that, appropriate tRNAs would be coming in, bringing the amino acids, and we would get translation of some protein, which would fold up into its characteristic shape right? And from this mRNA we would get 3 different proteins. We would get protein A, protein B, and protein C, coded for by gene A, gene B, and gene C. Now, as I said, these proteins would have some sort of related function, right? So whatever they're doing, the prokaryotic cell would need to have them all either expressed or not expressed at the same time. Right? So this is a way that prokaryotic cells can coordinately regulate or regulate at the same time genes that are doing a similar sort of function and express them all together or not at all. If we take a look now at how genes are organized in eukaryotic cells, so again, these would be things like single-celled organisms like protists or yeast, or multicellular organisms like animals and plants, what we would see is something completely different. What we would find is low gene density, and I'll explain what that means in just a moment, and we would also see that genes are, in general, not clustered together by function, and as a result, genes are not clustered by function and therefore are not regulated as one unit. In fact, they're independently regulated. So each gene is regulated as a distinct unit. So if we take a eukaryotic cell here, we know eukaryotic cells, in contrast to prokaryotes, have a nucleus and other organelles out here. And we know, we've talked about this before, that in the nucleus, in a cell that's not dividing, the DNA would be in this kind of diffuse fibrous form that we know is referred to as chromatin. It's DNA wrapped and coiled around proteins to organize it and compact it so that it fits is in an organized fashion within the nucleus. And we know what that chromatin actually is. It's made up of multiple linear DNA molecules wrapped around proteins and organized in a coherent fashion so that it fits within the nucleus. But if we zoomed in and we took a look at that genetic information, we would see something maybe that looks like this. We'd have chromosome, let's call this chromosome 1 here, chromosome 2, I'm just going to abbreviate that, and say chromosome 3. And we would find genes on those chromosomes, but in contrast to what we saw in prokaryotes, those genes would be spaced very widely. So a lot of intervening DNA sequence within individual genes. Now, in the human genome, we've got roughly 25,000-ish genes. And again, each one of these is one instruction to the cell for how to make one thing. Now, as we've talked about before, there are a lot of genes that are conserved between prokaryotes and eukaryotes, so genes that code for basic functions. So we might even have proteins A, B, and C, whatever they were, that we just talked about in the prokaryotic cell, but in contrast to what we saw in the prokaryotes, those genes would not necessarily be clustered together within the genome. In fact, they'd be scattered on different chromosomes, and each of these genes would be independently regulated. So if we zoom in on one of these genes, for example, let's just blow that up and take a look at what that gene structure would be. So this is gene C that codes for protein C. At the beginning of that gene, as with all genes, there would be a promoter sequence that serves as the binding site for RNA polymerase, so that part's the same. All genes that are expressed need a binding site that RNA polymerase can recognize as the beginning of the gene. Right? And so RNA polymerase would go along this gene, it would copy this gene, synthesize an mRNA, and that mRNA, just like what happens in prokaryotes, would be bound by a ribosome. That ribosome would translate that mRNA and we would get protein C as a result. Right? But now the difference here, in contrast to what we saw in prokaryotes, is that this protein is produced independently of proteins A or B and could be independently regulated, right, apart from either one of those. So you get more flexibility in terms of how the organism can regulate these genes. In prokaryotic cells, all the genes are either turned on or off. In eukaryotic cells, each one can be independently turned on or off, or, slightly tweaked to be a little bit more expressed or a little bit less expressed. Doesn't have to be completely on or off. Okay, so that's the basic idea between how genes are organized within the genome. Now let's actually take a look at how gene expression can be regulated in prokaryotic versus eukaryotic cells. As our model for understanding how transcriptional regulation occurs in prokaryotic cells, we're going to look at a set of genes that are collectively referred to as lactose utilization genes. And as that name implies, these are genes that code for things that the cell needs in order to use lactose. So what is lactose? Lactose is a disaccharide. We've talked about disaccharides before. Disaccharides are biomolecules that are made up of two individual monosaccharides. So we're talking about carbohydrates here. We spent a lot of time talking about metabolism and how cells use carbohydrates for energy, feed them into glycolysis. But before the cell can do that, it actually has to split this disaccharide into its monosaccharide components, so into galactose and glucose monosaccharides, which can both be fed into glycolysis, right? And we know what the cell gets out of glycolysis. The cell gets lots of energy in the form of ATP, right? So the cell has an enzyme that can catalyze that hydrolysis reaction, splitting apart this lactose into its two monosaccharide components. Not so important that you know the name of this enzyme, but if you're interested, there it is. It's an enzyme called beta-galactosidase. It splits lactose into two single sugars, galactose and glucose. All right, so this would be one gene product that the cell would need to express if it wanted to use lactose as an energy source. But if you think about it, there are others, right? In order for this cell, let's just draw our little boxy cell here. In order for that cell to get lactose that's outside the cell into the cell, we've talked about this before, that lactose isn't just going to freely cross through that cell wall and cell membrane. In fact, it would have to be specifically transported into the cell, and so you would probably need to have a membrane transport protein to carry out that function as well. Right? So these would be two gene products, right, coded for by genes within the E. coli genome that would have to be expressed in order for that organism to be able to use lactose as an energy source. If you think about it for a minute, the only time that E. coli really needs to express these genes at high levels is if there's lactose around, right? If an E. coli cell finds itself in an environment where it's some different sugar out here, let's say there's glucose or maybe even something else like maltose present, it's not going to do the cell any good to express this beta-galactosidase that specifically splits lactose into its component sugars, right? So if you're thinking about the logic of gene regulation from the standpoint of the needs of the cell, when would the cell actually need to express these genes at high levels? Only when lactose is around, not when lactose isn't around. So ideally, these lactose utilization genes would be regulated in such a fashion that they'd be turned on at high levels when lactose is present, but they would be repressed or turned off when lactose is absent. And that's exactly the way this gene regulation system works. So let's take a look at this in a little bit more detail. So how does a cell like E. coli regulate the expression of those genes such that they are expressed only when lactose is present and not when lactose is absent? Well, that involves coordinate regulation of those genes, as we talked about before. Genes with a common function are clustered in an operon, and these genes are no different. So those genes would be in some segment of DNA. Right? These are the genes that code for those proteins that are required for lactose to be used in the cell, and those genes would have a common promoter sequence. That promoter is the binding site for RNA polymerase to bind and sit down and transcribe those genes, right? So those lactose utilization genes are present within a cluster in a lactose operon. And now if we think about it, the cell has to basically be able to accomplish two things here. First of all, when lactose is absent, when there's no lactose around, the cell will repress those genes' expression, right? No sense expressing them if there's no lactose around to utilize. When lactose is present, the cell will activate expression of those genes. So we get those protein products and the cell can then utilize that lactose sugar. So how does it accomplish that? How does it repress these genes when lactose is absent? Well, that relies on a transcription factor that's called a repressor protein. All right, so this is a transcription factor, TF. That transcription factor is encoded by a separate gene in the genome. It's not part of the lactose operon, but it's what's referred to as a regulatory gene. Why? Because its protein product, the repressor protein, acts to regulate some other gene, or in this case a set of genes. So what does repressor protein do? Repressor protein binds to the DNA at a sequence within the lactose operon that's called the operator. The operator's sequence sits right next to the promoter sequence, which, as we said, is the binding site for RNA polymerase. When repressor protein is bound here at the operator, RNA polymerase can't bind to the promoter and therefore can't transcribe this set of genes. Right? Makes sense. Repressor blocks RNA polymerase from access to the promoter and can't get in there and transcribe these genes. Or, in reality, can only get in there occasionally at very low levels and transcribe these genes at very low rates. Okay? So they don't get transcribed very much at all. So how does this situation change then if lactose is present, right? We have to have some way of stopping this repressor protein from repressing RNA polymerase from being able to get in there and transcribe these genes. So how is that accomplished? Well, it's actually a very ingenious mechanism, right? This repressor protein is actually regulated by lactose sugar, right? When lactose binds to repressor protein, it changes the shape of that repressor protein in such a way that the repressor protein can't bind to the operator. So what happens if repressor protein can't bind to the operator? There's nothing blocking RNA polymerase from coming in here and binding, and I'm just going to abbreviate that RNA pol. So RNA polymerase can come in and bind to the promoter and can truck along down the DNA, transcribing mRNA as it goes, and we know that that mRNA is then going to be translated by ribosomes, and we're going to end up translating those proteins encoded by these genes, right? And remember that what these proteins actually do is allow the cell to transport and use lactose, right? So they're expressed only when lactose is present. Makes sense, right? So in that sense, lactose actually induces expression of these genes that are required in order for the cell to be able to use this sugar. So lactose is referred to as an inducer of the operon, but it does so indirectly, right? Recognize that what lactose is actually doing is binding to this repressor protein, causing a shape change. When it does, that shape change prevents this repressor from being able to interact with the operator sequence in DNA, so the shape change prevents it from being able to lock onto the DNA, and because it's not there, RNA polymerase doesn't get blocked, and we see transcription of these genes. The lactose operon illustrates several commonly observed features of prokaryotic transcriptional regulation. The first of which is the existence of operons, which, as we said, are those units in which genes that have related functions are clustered together and are regulated together, right? They have one promoter, and that promoter serves all of them. The second commonly observed feature that's illustrated by the Lac operon is in the way that the transcription factor, the repressor protein, regulates its expression. In genes that are regulated at the transcriptional level, very often what you'll find in prokaryotes is that only one or at most a few transcription factors are required in order to appropriately express that gene. And the other thing that we see with the transcription factors that act in prokaryotes is that they tend to have binding sites that are very near the promoter. And as a result of binding near the promoter, they directly interact and influence the ability of RNA polymerase to do its job, which is to transcribe a gene, right? Transcribe DNA to make RNA. As we're going to see in eukaryotes, things are a little bit different. So as we talked about earlier, eukaryotic cells don't cluster genes into operons. They do have transcription factors, but the way that these transcription factors work in eukaryotic cells is a little bit different from what we saw in prokaryotic organisms. So let's take a look at that now. Even though we're talking about the same basic process, regulating the rate at which transcription of a gene occurs, there's some important differences between how that's achieved in eukaryotic cells versus prokaryotic cells. So first of all, as we've already said, operons are not found in eukaryotic cells. So we don't see clustering of genes by their function. Genes are scattered throughout the genome, and they're independently regulated. So that's an important point. The second general feature that we see in eukaryotic transcriptional regulation that makes it a little bit different from what we saw in prokaryotes is that in general more transcription factors, and again I'm abbreviating that TF, are required for a given gene. So rather than just having one or two transcription factors regulating a gene as we see in prokaryotes usually, in eukaryotes we can have literally dozens of transcription factors that are interacting to regulate a given gene. Another thing that's different about those transcription factors is that rather than interacting right at the promoter, they tend to bind to DNA sequences that are far from the promoter. So here's a little cartoon diagram of a eukaryotic gene, right? There's the coding sequence of the gene. Here's the promoter of the gene where RNA polymerase would bind, and then the transcription factor binding sites can be up to tens of thousands of nucleotides away from the promoter. So they're really far from the genetic information that they're actually regulating. Just like we saw in prokaryotes though, these transcription factors, so the transcription factors, here's RNA polymerase. Now the way that these transcription factors interact with RNA polymerase, we know that RNA polymerase binds here at the promoter. If these transcription factors are binding here, the only way that they can interact directly is by a looping mechanism of DNA that brings those far away transcription factors in direct contact with this transcription complex, as it's called. So bending of DNA is commonly seen. And again, usually these transcription factors are activating transcription. And so when you have those transcription factors binding at those faraway sites and then looping around to contact RNA polymerase, that leads to activation and increased gene expression and more mRNA produced from that gene. So the way that transcription factors are interacting with the RNA polymerase transcription complex is a little bit different. But the basic idea is the same, right? They interact and that leads to a change in how that complex assembles at the promoter, whether it assembles at the promoter, and either activation or repression, right? But the other feature of eukaryotic transcriptional regulation that's really different than what's seen in prokaryotes, actually not found in prokaryotes at all, is that in eukaryotes, one way that we can regulate gene expression, and that's by regulating chromatin structure itself. All right, so regulating the structure that the DNA is found in rather than by direct interactions of transcription factors with the transcription complex, right? So we can make the chromatin more compact or less compact and therefore indirectly influence how easily RNA polymerase can access the promoter that it needs to find in order to express this gene.
So as we've discussed previously in this course, we don't actually ever see DNA in this double helical form by itself, what I call naked DNA. We never see that within the cell nucleus. Instead, what we see is DNA that is found in complex with proteins called histones to form this physical structure that we call chromatin. So chromatin is DNA wrapped around histone proteins and coiled up and compacted and condensed to some degree within the cell nucleus. And this allows the cell to fit a tremendous amount of DNA within the nucleus. It also provides a way for the cell to organize and regulate the position of that DNA within the cell nucleus as well. And this is really important for maintaining appropriate gene expression. Now, within the context of our discussion of gene regulation, what we need to recognize is that these different levels of compaction are associated with different levels of gene expression. And if you think about this, it makes sense why. I mean, if RNA polymerase is going to find, let's say this is a little promoter here that RNA polymerase needs to find. If RNA polymerase is going to be able to get access to that DNA, then the chromatin has to be in a pretty loose or open, right, open chromatin structure, less condensed state, in order for RNA polymerase to be able to access the promoters that it's looking for in order for genes to be expressed. If chromatin is in this more highly compacted or closed chromatin state, then RNA polymerase really can't gain access to the promoter sequences in the DNA that it's looking for, right? They're all kind of locked up within this physical structure of the chromatin. And we've discussed already in this class this most highly condensed form of chromatin that's ever observed, and this would be seen only during nuclear division. So either during mitosis or meiosis, you would see this most highly compacted form of chromatin present. Otherwise, you'd never see that, right? And the reason becomes clear if you think about it from the perspective of gene expression. When the DNA molecule is in this state, all of the genes in that DNA are silenced, right? They are not expressed. They can't be expressed because RNA polymerase has no access to those genes. It can't get in there. The structure of the chromatin is too highly compacted, too condensed. So in the normal life of the cell, it's the only time you'd see that highly compacted form of chromatin. But during interphase, we actually see a high level of variability of chromatin structure that kind of goes back and forth between this more open, less condensed, less compacted state of chromatin and this more closed, more condensed, more compacted state of chromatin. And this change in structure can be regulated by the cell in a couple of different ways. One way that this can be regulated is by modifying the structure of these histone proteins themselves. So what I'm talking about is chemical modification of histones, in which different chemicals are added on to the histone proteins, and that changes the structure of chromatin in one form or another. Now, this can happen in a number of different ways. The most common is by adding a chemical group called an acetyl group onto a histone protein. Acetylation is what this is referred to as, adding a little acetyl group, which looks kind of like this. You'd have a carbon double bonded to an oxygen and then also bound to a methyl group. So that's an acetyl group. And if you add that acetyl group onto a histone protein, then it changes the interaction of DNA with those histones. It changes the structure of chromatin, and actually leads that chromatin to become more open, and therefore to allow more access by RNA polymerase to the promoter sequences and more gene transcription. Now, an important thing to note about this is that this addition of this modification onto the histone proteins is reversible. So enzymes added on, other enzymes can come along and remove it, and by removing it would take the chromatin back to a more compacted state and decrease transcription. So this is a reversible modification at the level of the proteins that DNA is wrapped around to form this chromatin structure. Now, there's a second way that's commonly used in cells to alter the structure of chromatin, and this is a little bit more dramatic and long-lasting, and this is by chemical modification of the DNA molecule itself. So we're not talking about modifying the proteins here, we're actually talking about adding chemical groups in the form of methyls, these CH3s are methyl groups, onto the DNA molecule itself, right? And by adding those methyl groups on, that takes the chromatin into a more compact, more condensed state, and tends to repress transcription because of it. So more tightly compacted chromatin leads to repression of transcription. Now, in contrast to the histone modification that we just talked about up here, methylation of DNA is a more long-lasting form of changing chromatin structure. Essentially, this is irreversible. So cells would only use this form of regulation if they were really interested in silencing a gene for good, right? Now, under what circumstances would a cell find it appropriate to shut down a gene or set of genes for good? Well, this happens all the time in multicellular organisms. If you think about, for example, just a little skin cell, well, that skin cell is a skin cell because of the particular proteins that it expresses, right? So it expresses, I want to call them skin genes, right? These are genes, for example, genes for this structural protein keratin. It makes our skin cells a little bit tougher, a little bit of a better barrier between us and the outside world. But on the other hand, these skin cells would never need to express things like muscle proteins that allow muscle cells to contract. So these skin cells would silence these muscle-specific genes, would never need to express them ordinarily in the life of that cell. So methylation might be a good way for this skin cell to permanently silence genes that it will never need to express over the course of its life. Now, an important thing to note is that this modification, this change in chromatin structure, and its corresponding changes in gene expression. Now, the final thing to recognize about this form of transcriptional regulation, this kind of indirect form of transcriptional regulation that occurs, is that it is limited to eukaryotes. Prokaryotic cells don't have chromatin. They don't wrap their DNA around histone proteins and form this chromatin structure, and so they don't regulate genes in the same fashion.
Based on what you've learned in this video, you should now be able to answer these concept check questions regarding transcriptional regulation in prokaryotes and eukaryotes.